In this video, we're going to be looking at this lazy monkey, but also we're going to be talking about a very important topic called proteins, okay? So it's very important in the IB especially. Now, who's better to use than Cristiano Ronaldo? We know he's a human just like us, and he's made up of a lot of cells, right? Billions. And we thought that cells are the smallest things we're made of, right? The smallest living things, right? Now, what is this living thing made of? This living thing is made up of four fundamental basic structures, right? So it's the smallest living thing, but this living thing is made up of some other things. What are these things? Okay, it's four very important things: lipids, protein, carbohydrate, and nucleic acids. And this may jump to your mind and you realize, this is what I eat in my diet. I make sure to get some carbs, some protein, some lipids, and some nucleic acids. Maybe not this one, but this one is basically DNA. Nucleic acids is what what makes up DNA. And when we eat other organisms, we eat their cells, right? Basically. So when we digest their cells, we'll also need to digest their DNA and all that, right? But you basically think of these three important ones. Now, why do we eat these? We eat these because we want to make sure our cells can be made properly. Our cells are made up of these four things, and if we don't eat them, how can our cells make them and use them? I mean, how can our body make proper cells if we don't ingest them, right? Now, the important one for this video is proteins, okay? We know lipids, carbohydrates, they typically serve as energy, right? We eat them so that we can break down and make ATP so that we can move around and keep doing things every day. Protein doesn't really serve that purpose. It has an insane amount of other reasons, not really energy. When we don't have any more lipids or carbohydrates, so when you're starving, then your body might go to protein and break that down, which is why you'll lose muscle and become skinny if you starve yourself. Okay, but protein, in reality, does a lot of things. The one thing you may think of is muscle, right? You think of athletes as eating a lot of proteins, and that's why they're so strong. That's partly true. One of the functions of protein is muscle. They help make your muscle, they help make you strong, but they have an insane amount of other, probably more important functions, okay? So let's continue. So here we go, protein. Once again, first off, you need to know how exactly proteins are made. So proteins is coded for in your DNA. Your DNA is the instruction manual inside your nucleus that will tell your cell exactly how to make proteins, okay? Because you have an insane amount of them in your body, and they all look slightly different, which is why they all have a slightly different function. So, your cell will read this DNA using something called ribosomes. These ribosomes will read this DNA, and after it reads it, it's going to know how to make the protein. So it's going to use the instruction manual to make a protein. So that's basically how it works. So your DNA is what tells your cells how to make specific proteins that the cell needs. Like different cells will need different proteins. So muscle cells might make muscle proteins. Other cells will make other kinds of proteins, okay? So not each cell will make the exact same protein. They'll make some different ones. Now, let's say this will represent our average protein, okay? This represents all proteins. I'll give you some examples here of important proteins. Insulin. We know insulin is very important. It reduces your glucose in your body. For example, you eat too much sugar, you just had a massive cake. That's pretty bad because if your glucose goes too high in your body, you can have serious problems. You can die. That's what happens with diabetic people. They don't have this protein, and so their glucose level after they eat gets so high that they die. So this one is very important in making sure our blood glucose level is at the reasonable level. Immunoglobin functions as a protein that helps us keep healthy. Okay, it helps us fight infections. Collagen helps our bones stay strong, and our ligaments and our cartilage and all this. Okay, it's a structural thing. So both of these had a functional thing, and this one was structural. It helps for stability and strength. And enzymes, this is a big category. There are many enzymes in your body, and enzymes can do many things. make many reactions happen. I made a whole video on just enzymes, so don't worry too much about this if you don't understand it right now. And spiders also make a protein. You know that annoying spider web? That's made of spider silk. That's also a protein. Okay, so based on this, we can see that proteins are not so simple. They have so many functions. It's not like a protein does one thing. A protein has an insane amount of functions. And you can kind of think of Bob the Builder. Proteins kind of remind me of Bob the Builder. They have so many jobs in our cells. They fix things, make things strong, make things happen. In addition to making things strong and all that, and making things happen, catalyzing many reactions, they are also structural. They can just simply be there without doing anything, but in being there, they're strong. Okay, so many functions, just know that, okay? There's not one kind of function a protein can do. Okay, now let's get further into how they're made. So first of all, I just told you that your body, your cell will read the DNA, it will read this instruction manual, and using ribosomes, it will translate or convert this instruction manual and read the steps and make a protein. Now, these ribosomes are not in the nucleus though. They are in the cytoplasm, okay, outside the nucleus. So we know that proteins are made outside of the nucleus, inside the cytoplasm, the little blue space, okay? So let's go there. So inside, let's go around here. Inside the cytoplasm, we're gonna have a whole bunch of amino acids. These are little balls, little structures, okay? And these will eventually make proteins. So they're floating around. Okay, and you have about 20 different types of amino acids in your body, 20 different types. And depending how you arrange them in different orders and how long you make them, that will make different proteins. Okay, so you can imagine if you have 20 different amino acids, you can make an infinite amount of combinations and therefore an infinite amount of proteins, right? So we have these amino acids, and they're floating around. Okay, in the cytoplasm, right here. Now these ribosomes will grab these amino acids according to the instructions and put them together to form a protein, okay? So here we have some amino acids. The DNA will be read, the instruction manual will be taken. The ribosomes, as I mentioned, will grab these amino acids according to the instructions and put them together into a chain. This chain is called a polypeptide. Poly means a lot, means many, and peptide means one of these things, okay, like an amino acid. So you have many of these amino acids put together, okay, it's a chain, it's like a chain. So here they're separated, but here they're put together in a specific order according to the instructions of the DNA. Now, this is also known as the primary structure of a protein, okay, the primary structure. It's the basic structure, because proteins are basically, in the simplest definition, a protein is amino acids put together. Therefore, this is essentially the simplest form of a protein, the primary structure, okay? Also called a polypeptide. Now, it can get slightly more complicated than this, okay? So we just put it together into a chain, but a protein can, some changes can happen to this polypeptide to make it into a more specific protein, because right now it's just a chain with a specific sequence of amino acids. So let's get into it. So this polypeptide can basically fold itself into a specific structure. For example, one important one you need to know is a helix. Do you see this? It's like a helix shape, okay? You can see. So it forms this funny structure, and we're going to see exactly how this happens and why this happens later. But this is called still a polypeptide because it's still a chain of amino acids, but it's now called the secondary structure. So here it was primary, it was just a chain. Now that it's folded in a weird way, specifically the alpha helix, is called a secondary structure. So this is one interesting way that this polypeptide can be converted into. And another one you need to know is called a beta pleated sheet. Okay, and it looks like this, kind of like stairs. Okay, so they fold like this into stair shapes. Do you see that? Okay, so again, we're going to talk about this one also. So these are both secondary structures. Bear that in mind. So primary is the straight chain. Secondary is the weird little things they fold into, either an alpha helix or a beta sheet. And we'll talk about them later. Now, this is not where it stops. These can now combine. So for example, say the DNA made you make one coded for a polypeptide that turned into this, and then another piece of the thing, another piece of the code made the cell make a beta-pleated sheet. Now you have two of these secondary structures. If they combine, you form a tertiary structure. So you can see here we have a bunch combined. We have two alpha helixes combined with two beta-pleated sheets. So this is just random. I'm not giving you a specific real example. But so basically, a tertiary structure is when these secondary structures combine together. Okay, so that's what a tertiary structure is. Now, does it stop there? No, there's one more stage to a protein. A protein can have a final form, which is called a quaternary structure. And this is basically when a bunch of these tertiary structures come together to form a polypeptide. Okay, so you can make a bunch of different shaped tertiary structures, and when they combine together, they'll form a very interesting, complicated, intermingled shape, and this is a quaternary structure. So remember the protein we had before? This is the one we just made. Look. See? It folded. This protein we made was specifically this structure, and this protein will have a specific function based on its structure. So that's what you need to know about how it's made. Okay, we're going to go in a bit more detail that you need to know, but this is the overall idea. You have amino acids, your ribosomes will read this DNA, this code, and based on that code, it will make a polypeptide. This polypeptide will then fold into different secondary structures, either an alpha helix or a beta-pleated sheet. These different alpha helixes and beta-pleated sheet can combine together in different ways to form a tertiary structure. And then these tertiary structures can combine with each other to form a quaternary structure. And this is a protein. This is how proteins are made. Okay? Now, let's get into a bit more detail. This is the overall image. But when I said these amino acids are combined together, how exactly does that work? Let me show you. So here we have a polypeptide, okay, a chain, and I'm going to show you how a structure of the amino acid looks like. So we're zooming into this little amino acid here. You can see it's really nice, this round amino acid, and this is what it's made of. This is what the structure of all amino acids look like. Do you see here? We have, don't worry about this. This might confuse you if you don't know any chemistry, but don't worry, you don't need to know chemistry. There's just some basic things you need to memorize in that case, and it won't come up on your test. Any of these chemistry related, you don't need to understand this. You need to remember how it looks like and all that, but understanding what's going on here is not too important for a biology student. It won't come up on your test, the understanding part. But I'm going to explain a little bit to make it clear to what you need to know. So this is some elements: nitrogen, carbon, oxygen, hydrogen, and this is called an R group. Okay, and we're going to get to what that means just now. So this is a molecule, okay, a molecule, and all amino acids look like this. The only thing that's different in amino acids, in between different amino acids, is the R group. The R group. So this R group doesn't mean anything. It doesn't mean like an element. This means a group, meaning this R can be anything. Okay, it can be a bunch of carbons, it can be a bunch of oxygens, it can be a bunch of other elements. Okay, so this R group just means kind of unknown. That's what it means. It's an unknown thing. So this amino acid may have a different R group from this amino acid, or this one, or this one. They will all have a different R group, but everything else will be the same. The whole other thing I'm showing here will be exactly the same. So the only way in which they differ is this group here. So let's get into some information. We need to know some more structures here. We know this is the R group. This is the thing. This is the group that distinguishes and makes the amino acids different. But what is this group here? This N, this nitrogen, this hydrogen, this hydrogen. Okay, this is called the amino group. Amino group. Amino group. Okay. Again, you don't need to understand anything here. You just need to remember this. So I'm trying to make it as easy as possible for you to remember. So this is an amino group. Then we have a carbon and a hydrogen. They're not labeling this for you, but they care about this group here. This carbon, oxygen, hydrogen, okay? This right here is called a carboxyl group, okay? Carboxyl, let me put it here. So you need to know these two groups. This is an amino acids. An amino acid is the smallest unit of a protein. A bunch of amino acids put together will form a polypeptide, and all this will happen. Now, an amino acid has this structure. It has an amino group, a carboxyl group, an R group, okay? And then a carbon and a hydrogen here, okay? These are the three important groups that you need to remember. But you need to remember that the R group is the thing that distinguishes them. Now, you notice all of these lines in between these carbons and hydrogens and all these elements. These represent bonds. Okay, so this carbon is holding on to this carbon, and this carbon is holding on to this oxygen, and so on. This bond is called a covalent bond. So I'm going to put it here: covalent bonding. These bonds are extremely, extremely strong, meaning they won't be broken. They are almost impossible to break. Okay, very difficult to break. Covalent bonding. Now, this is also, one amino acid is called a monomer, okay? Mono means single, right? So it's a singular. It's like a single person, okay? Now that we know what one amino acid looks like, the structure, how do we combine one amino acid with another? Like how is the bond made? How is the interaction made? So here we go. So here we have the same polypeptide. Here we have this one. And then right next to it, we have this color, right? So how did these two combine together? So here we have the structure. See, it's the same. Everything is the same between this amino acid and this amino acid. The only thing that is different is the R group. You notice I showed it by changing the color. They will have a different R group. Now, how do they combine together? Let me show you. So the carboxyl group of one amino acid, so this group, will interact with the amino group of the other amino acid. So let me show you. So for example, the OH group from the carboxyl group of this amino acid will interact with the hydrogen of the amino group of the other amino acid. And what's going to happen is, what do you see? I see H, I see two H's and an O. And we know H2O is water. So when these two amino acids come together, they will combine together, and in the process, they will release this water, this H2O. Let me show you. Let me move this. So this will pretty much just pop off. The water will pretty much come off. You don't need to understand the mechanism because that's chemistry. You need to remember that when they come together, a condensation reaction happens. A condensation reaction is when water is lost from the reaction. So water will be lost as they combined. So water will get removed. So it's a condensation reaction. And in the process of water leaving, a bond will be formed. And this bond is called a peptide bond. Okay, so let me show you. So here, we can see these two. Let me show you. So let me show you here. I'm going to separate them. So remember, I said the OH is going to come off, right? And that H is going to come off. And when they come off, they're going to combine to form water. Now that they've come off, there's a bond that this nitrogen wants to make. And this carbon also wants to make a bond because now it lost one of its bonds. So they will come together then to just share that bond, okay? So they both lost a group, and now they had an empty space to hold a hand with, and now they're holding hands, they're binding together. And this bond right there is called a peptide bond, okay? Peptide bond. I tried to make it glow a little bit so it's clear which bond I'm talking about, okay? Again, this is pretty confusing if you don't understand chemistry, but it's just something that you may unfortunately just have to remember because they do ask you to draw this. Now, so remember how I said one amino acid is called a monomer. So when you combine one amino acid with another amino acid, like I just showed you, now you formed two amino acids together, and this is called a dipeptide, okay? Or a dimer. They call it a dimer or a dipeptide, okay? Because remember, polypeptide is when you have many. Dipeptide is when you have two attached to each other. Now again, this process can be repeated. So we just put two together. Now we can put three, because here we have the two from before. Now this OH will interact with, oh, by the way, it doesn't matter which H is lost from the nitrogen. It can be either. So for example, we can now show how this will happen. Instead of these two, instead of this one and this one, we can say these two will now interact. and now a water will be lost, okay? And that will, in the process, form another bond between nitrogen and carbon, okay, called a peptide bond, okay? And now you'll have 3 amino acids in a row. So this process will just repeat forever. And at the end of the day, whenever you have a chain of amino acids, the one side will always have an amino group, and then at the other side you will have the carboxyl group, okay? So it's always going to be like that. Now that we know how these amino acids get converted into a polypeptide, now that we know the structure and how this bond forms, we're going to take a little bit of a look of how they get turned into these specific secondary structures, okay? Because they don't just fold magically out of nowhere into these structures, okay? There's got to be a reason why they form these structures. There's got to be something that makes them come into this specific shape. So what is that? So the first one we'll look at is alpha helix. So the reason why an alpha helix shape is formed is because of this, okay? Here we have 2 amino acids put together, a dipeptide. So I'll just put it around here, okay? So notice, remember, here's the bond. Let me show you clearly. This is the dipeptide bond, right? The peptide bond. And here's the nitrogen with the hydrogen on it, right? We're looking basically at this spot here, right? To show this needs to be clear. And then I'm just filling in some of these amino acids to make it clear. And then we have another one here, right? And this happens everywhere. It's the whole way through. I'm just showing these two for the sake of this explanation. But so I'm going to move them now here. Okay, let me put them here. It's hard to see on this diagram, the 3D, so I'm putting it the exact same thing on this side so you can clearly see what I mean. Okay. Okay, look at this. Look at this, very important. So notice something. So this hydrogen here on the nitrogen is going to bind, has a very big attraction for the oxygen here on the 1 carbon, okay? So they will interact with each other like this. They have a nice pull, so I'm going to show it here. Do you see they're right above each other on this helix, and they have a really strong attraction for each other, okay? This hydrogen has a very strong attraction for the oxygen here, okay? And this is why all around this helix, all the hydrogens from above, from the layer above, will be attracted to the oxygens of the layer below, which is why this helix forms. And this is called hydrogen bonding. Again, you don't need to understand the mechanism. Just know that alpha helixes form when the polypeptide chain folds into a helix due to the hydrogen bonding, the attraction between the layer on top with the layer on the bottom. The hydrogen from the layer on top with the oxygen from the layer at the bottom. Because but you need to know one thing. Remember how I said before that, let me show you here, that these bonds here are covalent. They're very strong. They don't get broken. Now, these bonds here, in between, forming the being the reason for the alpha helix, these are very weak. Okay, so hydrogen bonds are much weaker than these covalent bonds. And this will be very important later in this video. So that's how an alpha helix forms, and I hope this kind of makes sense. Again, I don't want to give too much detail because it's not the level you're required to know, okay? But this will hopefully give you some idea of why it falls into the shape. Okay, this is called hydrogen bonding. Now, how about beta sheet? So a beta sheet looks like stairs, right? So to understand this one, I'm going to show you this part first. So here we have a polypeptide. A beta sheet essentially forms when this polypeptide has for some reason, so these amino acids down here have an attraction for the amino acids up here. So what happens is it folds. Okay, so this top part will come and fold in half like this. And when it folds in half like this, it folds in half like this because these amino acids are attracted to each other. So there's bonds forming in here. Okay, so let me show you. So here I have it. So remember, this was initially one long chain, and now it folded in half like this. And the reason why is because these amino acids are attracted to each other and want to make bonding in between, just like this. Let me show you here. Again, here we have a dipeptide. So here, two amino acids attached together. And again, we know that this bond here, this peptide bond, has a nitrogen, is between the nitrogen and the carbon, and it has a hydrogen on it, remember? And then this layer here, next to it, that was folded, has this oxygen. So again, the same thing as the alpha helix, only in a slightly different orientation. There will now be this bond. Let me show you. Let me move this bond. This bond between this oxygen and this hydrogen, just like in the alpha helix. And if this keeps happening all the way down, that's why this beta sheet is forming. So this beta sheet is pretty much one row of amino acids, and then that row of amino acids is folded in half and folds down and interacts with its own polypeptide. So that's a beta sheet. And this bonding is also hydrogen bonding. And remember, it's slightly weak compared to covalent bonding. So that's now we've covered officially the secondary structures, why they kind of the reason why they arise. But again, we don't stop there. So we got to keep moving. So now, how does this tertiary structure form? We know now how these alpha helixes and beta pleated sheets form. So what about the tertiary structures? The tertiary structures form, right, when these alpha helixes and beta sheets combine together. Now, what causes them to bind together? What causes them to interact with each other? What causes these bonds to be formed? So these bonds that formed are specifically between the R groups. So remember how amino acids, let me go up here, have this R group, right? We saw that hydrogen bonding happens between this H group and this oxygen group from different amino acids, right? Now, this tertiary structure happens because the R groups are interacting with each other. So for example, let's go down here. So let's say this alpha helix has a bunch of amino acids, right? And they all have R groups. So for example, maybe this R group from that alpha helix likes to interact with this one here, and then maybe the R group from that helix likes to interact with this beta pleated sheet R group here. So all of these R group interactions are called, by the way, they're called disulfide bridges or ionic bonds. They pretty much mean similar thing. So in a nutshell, the thing that makes secondary structures, so beta pleated sheets or alpha helix, is hydrogen bonding. The thing that makes tertiary structures is R group interactions between these secondary structures, okay? So I hope that makes sense. Again, you don't need to understand the mechanism exactly why, you just need to know this fact. So let's get to the last part. We're almost there. Just be a little bit patient. So now we get to denature. So we know now how these proteins are made. We've gone now through everything. We need to make sure that we understand this part. So Ronaldo has a bunch of proteins in his body, right? And we know now how they're made. And they all have different functions. But we need to make sure that they always have good environment. Because if they don't have a good environment, they will turn out badly. They will break or get denatured. That's what we call it. So when a protein breaks, we call it denature. It's very important. It's like raising a kid. Well, your kid, if it has a good environment, it will be able to work well and become successful, just like this protein. But if this protein has a bad environment, and by bad environment, I mean high temperatures, high pH, many other conditions. If these conditions aren't good, we're going to denature this protein. Okay, what do I mean by denature? By denature, I pretty much mean reverse the process. So remember, we know now how these proteins are made until this final structure, but denature is the reverse structure, the reverse process, turning this protein back into what it was made of, okay? Reversing the process. Let me show you. So here, denature would be, say the temperature is too high. Remember what made this 3D structure and what made this protein fold like this? It was a combination of hydrogen bonds and ionic bonds, right? As I just mentioned. remember, these bonds, the reason why I made them like striped and not continuous is because they're weaker. So temperature, since they're not that strong, if the temperature goes a bit high, then these weak bonds can be broken, and so we can break this protein into its secondary structures. Now, the high temperature can then break these hydrogen bonds that keeps them in this alpha helix or beta pleated sheets, and this will then turn them back into a normal polypeptide. But the temperature doesn't break these amino acids apart because they are held together by a covalent bond, and a covalent bond is strong. Now, why is denaturing bad? Denaturing is bad because now we don't have this structure that we had before, this useful structure. Because with this structure, with this specific structure, this protein has a specific function. And if we break this protein back into its original polypeptide, that's different structure. And so this polypeptide won't be able to do the right function, okay? It won't be able to do the right thing. And so your body might slowly die, okay? So that's what denature means. And many things can cause this, okay? Let me show you a nice visual. So remember before we showed that this protein was made up of many tertiary structures. I showed you the image above. Denature would pretty much basically melt it. The structure would completely change and it'd be useless. Now, last final thing, two vocabulary words, fibrous proteins and globular proteins. So proteins, as I showed you how they're made, they can be made just as I explained above. Now, depending on the final shape the protein has, we give it a different name. If the final protein is more flat, like this one, we call it a fibrous protein. If it's like a globe, like a tennis ball, or like a ball, like 3D, then we call it the globular proteins. So this depends on the final shape of the protein. And we care about this because globular proteins tend to have specific functions, and fibrous proteins tend to have specific functions. So let me show you what I mean by this. Look. Fibrous proteins, so if they end up being flat in structure, they tend to be related to structure. So this one might be collagen, as I mentioned before. It might be used to make your bones strong or things like that. So here's some example. Yeah, collagen, this is bones, this is your hair, this is muscle. All these things are structural things. So fibrous proteins tend to be structural. And you don't need to worry too much about this, but they are mostly insoluble in water. So they will not dissolve in your blood well because they're insoluble. Your blood is mainly water. Now, globular proteins have different functions. They aren't really structural. They're rather functional. So they are like little workers. So instead of being a structure, they will do little things for you. For example, it could be hemoglobin, which is, we know this is a protein that carries oxygen around in your body. It could be insulin, which we know is a little worker that lowers the glucose in your body. It could be immunoglobin, which is a little protein that protects you from infections. Okay, so these overall, depending on their final structure, whether they're globular or fibrous, they will have a slightly different. They will be intended for a slightly different function, either structure or functional. So, and this one is mostly soluble in water. Again, this doesn't matter that much for you. So this is everything that you need to know about proteins. I know it's a lot, but I hope I helped you to understand stuff a bit, because if you can understand it a bit better, it makes much more sense. But again, there's some details that you may not get, and this is not your fault or anything like that. For biology, IB, you simply don't need to know it. So I don't want to drill it into your head because it's unnecessary. It won't show up on the test. So everything I taught here, you definitely need to know. So I hope this video helped you out.